So we'll leave it just a few minutes for people to log in, and then we will start, and then people will just continue joining. So Ed, do you spend most of your time at Georgia Tech or Emory, or you split it? How does it work?
Mostly Georgia Tech.
Mostly Georgia Tech, yeah.
Although I have been spending a reasonable amount of time at kind of the Emory VA these days with my, based on some of the collaborations we have in rehabilitation sciences.
Okay, okay. Yeah, I saw something when I was, like Googling what you have been doing lately. I mean, so I'm never sure because, like these two places are pretty far apart, but you guys still manage to spend time at both of them.
Yeah, it's the classic, 10 to 45 minutes away.
Yeah. Not too bad. Not too bad. Okay, so I think we can start. Welcome everyone to today's Tissue Talks. Before we introduce our speaker today, I would just like to remind everyone to please post your questions. You can do it throughout the talk using the Q&A feature, not the chat feature on the screen. And then we will answer as many questions as possible after the completion of lecture. It's my real pleasure to introduce our guest today. So this is Ed Botchwey from Georgia Institute of Technology. Ed received a very diverse education and training. So he first earned his bachelor's degree in mathematics from the University of Maryland. He went on to get a master's degree in engineering and material science from UPenn and stayed at UPenn for his PhD that was in bioengineering. Following PhD, he did a postdoctoral training at the Wistar Institute in Philadelphia. And shortly after, he was recruited by the Biomedical Engineering Department of the University of Virginia in Charlottesville as assistant professor. And he was then promoted to associate professor at the same department. This is the time when he moved to the Biomedical Engineering Department at Georgia Tech and Emory University. This was in 2011. It is also affiliated at Georgia Tech with the Marcus Center for Therapeutic Cell Characterization and Manufacturing, with immunoengineering, and with the Center for Immune Engineering. His laboratory is pursuing a highly multidisciplinary approach towards improving tissue engineering therapies, and they do this through studies of microvascular remodeling, inflammation resolution, and effects of the host stem cells, and really recruiting and utilizing the host stem cells towards tissue regeneration. Their overall goal could be summarized as the development of new and effective strategies to repair, replace, preserve, or enhance tissue and organ functions. Looking into their publications, their most influential papers are reporting development of bioreactors for engineering of bone and other tissues. Among many other recognitions, Ed Botchwey was elected to the American Institute for Medical and Biological Engineering. He was credited for significant contributions to immunoregulatory biomaterials and also to the national leadership in biomedical engineering in the design of materials with capacity for immune regulation and to quantitative studies of mass transport in tissue engineering scaffolds. And as you can see here, Ed will speak today about modulating bioactive lipids for regenerative engineering and measuring immune modulatory potency of mesenchymal stromal cells. Ed, thank you so much for accepting to speak for us today. So you can now take over the control of the screen.
Thank you so much, Gordana. That was a fantastic introduction. I don't know if I can kind of, whether my talk will be worthy of it, but I'll do my best. It's a real honor to participate in this series. I've been kind of blown away at the lineup of speakers that you have made available to this community, and I'm really excited to be among them. I am going to experiment on you a bit and talk about some things that may be, if you are familiar with my work, may be less familiar to you. And that is, I want to start by talking a little bit about sickle cell disease. I want to transition from there to talk about cell therapy manufacturing, in particular, mesenchymal stem cell manufacturing. And then I want to end things up in a way that envisions biomaterials that can begin to re-educate and re-engineer immune responses to promote musculoskeletal healing and repair. And I'm hoping that I can maintain a consistent theme about why these three topics and projects fit together in the same lab. So in a few days, we'll be commemorating Juneteenth, which is a holiday that we commemorate the ending of slavery in the United States. We're hoping that it can be made a federal holiday. I think the Senate just voted unanimously yesterday or the day before to make it a new federal holiday. And in the wake of the Black Lives Matter protests we saw last summer and all of the things that kind of remain current events, it's really been an inspiration for my group to begin to venture out of some of my comfort zone and traditional areas of research to think about ways in which, you know, our skills and knowledge as biomaterials scientists and biomedical engineers can potentially begin to address problems in other areas. And sickle cell disease, as a person like myself with a West African ancestry, is one that I am personally familiar with because of its impact on my family. But one need only look at the current relatively recent headlines in the esteemed New York Times to begin to appreciate the impact that a disease like this one is continuing to make, and specifically making on people with African ancestry. And I just, you know, this quote I use just as a motivation to consider the potential impact of breakthroughs in this area can give some relief to people who are essentially saying that the pain associated with the vaso-occlusive crisis and sickle cell disease manifestations is like an agony, like knives stabbing at the bone. We only have a couple of drugs for treating sickle cell disease right now. That's hydroxyurea and L-glutamine, sold under the name Indari. Bone marrow transplant is a potential curative therapy, but has obvious risks, and there are inherent difficulties at finding matching donors. Of course, with the advent of cell therapies and gene therapies, we can envision other much more exciting and innovative cures to treat sickle cell disease. But given the anticipatable costs of these kinds of therapies, we're going to be dealing with the impacts of sickle cell disease in adult populations for some time to come. And we need to envision therapeutic strategies that can be continued to address some of the disease manifestations, particularly the pain, but also some of the end organ damage that's associated with it. And with that in mind, I wanted to talk specifically about what we call sickle bone disease, which may be also unfamiliar to many. But this is a group of disorders that includes osteopenia, osteoporosis, and osteonecrosis that can occur and does occur quite frequently in adults suffering with sickle cell disease. It can result in collapse of the femoral head. It can contribute to bone pain and just the overall impact of sickle cell disease on quality of life and longevity. And the underlying molecular mechanisms of how point mutations in the hemoglobin gene manifest itself in inflammatory or otherwise bone loss phenotypes in sickle cell disease is not a topic that has been explored in great detail. Now, I am going to try to maintain a theme here that will require you to have some working familiarity with lipids in particular, right? So I'm not going to talk about proteins and nucleic acids as maybe kind of the more conventional molecules that many of us deal with, but lipids. And I'm going to talk about a particular class of lipids called sphingolipids. And this is just a diagram that I really stole off the web from a talk given by Rick Proya at the NIH, where he was likening the processing of precursor molecules in kind of the de novo synthesis of sphingolipids through to the production of so-called bioactive lipids and other more structural sphingolipids that are kind of present in plasma membranes and serve to determine kind of their mechanical properties. He kind of described this whole mechanism and network as like the metro in DC, because he lives and works in the Bethesda. But suffice it to say that these colors kind of represent different metabolic species that can kind of transition through a series of enzymatic reactions that can result in, I'm going to say, two major classes of sphingolipids that will be important to kind of think about in this talk. One are the complex sphingolipids, particularly the ceramides and the sphingomyelins. They're important in the nervous system. I'm not going to talk about that as much, but their roles in plasma membrane integrity and maybe some immune functions, if I get to it, are really, really critical. And you can kind of appreciate how this comes about just by looking at the structure of these molecules and understanding that based on the head group, these molecules can occupy different amounts of space in the membrane. And so they're essentially charged with maintaining kind of the sorting and trafficking of content in the plasma membrane. And they also have a role in the trafficking of intracellular vesicles and the secretion of exosomes that I'll talk about. They also have a significant role as signaling molecules, if I can, yeah. So along this corridor here, right? So the yellow and the brown line from the ceramide hub here. So ceramide converts to sphingosine, converts to this kind of charmed molecule called sphingosine 1-phosphate. I say charmed because it's one of these bioactive sphingolipids in that it can directly control cell functions through the activity of 5 known G-couple receptors that are creatively kind of labeled S1P1 through 5. And these are associated with and regulate a large number of cell functions, most cells have these receptors, and they're expressed at various combinations. And there's an interesting and complex biology underlying it. But what I wanted to build off of is that several years ago, we decided to use what we were learning about how to profile for lipids, particularly membrane lipids in cells, using high throughput mass spec. We decided that it was reasonable to expect that the shape distortions and alterations in the plasma membrane of red blood cells would very likely change the underlying metabolism of sphingolipids. And if that resulted in changes in bioactive lipid concentrations, that would really feed forward to a whole cascade of kind of chronic inflammatory stimulation. And that was really worth looking into. And so we did. Essentially, what you're looking at is a very brief summation of a paper we published several years ago now, where if you look at the density of the plasma membrane in red blood cells, and particularly you look at the highly distorted plasma membranes from red blood cells, you can see that this enzyme, this acid sphingomyelinase, so this is an enzyme that converts membrane sphingomyelin into ceramide, the activity in blood, red blood cells from sickle cell human cells, and in addition to, if you can kind of look in the blood of mice, you see that the activity of that enzyme is significantly higher than in a normal genotype, where both normal hemoglobin genes are in the what we call the trait phenotype, where you have one normal hemoglobin gene and you have a recessive sickle mutation. So that was interesting. And so, we wanted to know whether or not enzymatic reactions that convert plasma membrane sphingolipids, whether it actually results in the production of some of these pro-inflammatory metabolite species that are quite well studied in the literature of sphingolipids. And so we did kind of a broader profiling of sphingolipid distribution in the plasma membrane of red cells by mass spec. And so, here's just kind of a schematic of all the different structural variations that that determine essentially the members of the sphingolipid family. They have this kind of backbone sphingosine molecule here, but based on the head group or whether this fatty acyl chain remains intact. If it does, that's a complex sphingolipid, two acyl chains. If it's cleaved, those are called so-called long chain based sphingolipids. And based on the head group, you can get different structural species, and some of the actual substituents in this basic schema are listed here. And you can kind of see the difference between sphingomyelin and ceramide as an example by the selection of the head group here. But all of that can really be distinguished in a mass spec profile. And, for us, we wanted to look at both classes of both the complex lipids that are dually acylated and some of the long chain bases as well. And we wanted to particularly know whether, what are the concentrations of this regulatory molecule called sphingosine 1-phosphate, because that's the one that could really begin to alter and dysregulate immune homeostasis. And we thought that ultimately could be one of the root kind of molecular bases for something like sickle bone disease. And there's more reasons that we thought that, but I want to cover, I want to get to, some other parts of this story. But this part of the story culminated in a partnership that we formed with the Sickle Foundation of Georgia, where we were able to recruit donors, both in a normal genotype AA cohort, in addition to kind of a sickle cell cohort. And we selected a slew of individual sphingolipids, and I don't expect you to remember all of these complex names, but suffice it to say that all of these molecules that you see here are structural variants on that schema I showed you before. These are sphingolipids, and we essentially use mass spectrometric profile their concentrations in whole blood and the red blood cells themselves and in the plasma. And here's just kind of a summation of what we found, right? So sphingolipid profiles are very different between the SS genotype, people living with sickle cell disease, and the normal AA genotype. And we've done a variety of different methods of analyzing this high throughput data in an effort to really zero in on the things that are most different between these two profiles. And the main thing I want you to take away, so I've given you quite a bit of background on, that gets into some of the weeds of lipid metabolism and the molecular structures of sphingolipids. But if you look inside this green circle, you can see that in this is what's called, a volcano plot where we're just, we're graphing the essentially the magnitude of the difference along the x-axis against the log base 10 of the p-value to really identify the standout metabolites that contribute to the variance between an SS profile and an AA profile. And what you see is that things like sphingosine and this molecule S1P show up in the profiles as really being able to distinguish AA and SS. That suggests that some things can be going wrong here related to S1P signaling in people living with sickle cell disease. And do we have a basis for believing that that could have a significant role in what happens in sickle bone disease in particular? Well, I'm just kind of summarizing a number of studies that you can kind of find in the literature, and that S1P plays a role in the migration of mesenchymal stromal cells that can be the osteoblastic or osteogenic bone forming cells that maintain bone homeostasis. The differentiation from adult stem and progenitors in the bone marrow to osteoblasts or bone forming cells is also partially regulated by the concentration of S1P. Not sure why that's showing up so blurry, but the take-home message is this that you could barely read here is that if you look at animal models where S1P signaling via its receptors is dysregulated, you can see changes in the formation of bone. And also on the osteoclastic side, S1P has a significant role in terms of just the number of osteoclasts that appear in the bone, in addition to their activity. Cathepsin K in particular is the primary kind of cathepsin, or really the primary protease that osteoclasts use to resorb bone. And so we see that both on the anabolic side when bone gets formed and on the catabolic side when bone gets broken down, we're seeing that S1P concentration can play a role in regulating all of these things. And if that's true, could the changes in S1P concentration that happens in sickle cell disease be a root cause of what happens in sickle bone disease? Well, we wanted to see whether in a humanized mouse we didn't develop this mouse, but this so-called Towns mouse is a model of sickle cell disease, a preclinical model of sickle cell disease where the mouse hemoglobin genes have been knocked out. The human hemoglobin genes have been knocked in a way that you can replicate the 3 known genotypes that exist in humans, right? The normal AA genotype, the AS genotype, which I mentioned is what we call sickle trait, that is in some circles considered to be without a real disease phenotype, and then the actual sickle cell disease, where you get both of these, where you have both sets of sickle genes. And so in the mouse model, we can determine, we can generate these different phenotypes, and then we can use micro-CT in order to determine whether some of the hallmark features of microstructural changes that happen in bone are also occurring in the sickle mouse. And that would give us a basis for study, as you might imagine. And to make a long story a little shorter, because we're hoping this paper is going to come out soon, the answer is very much yes. You can look across a broad spectrum of microarchitectural features by microcomputed tomography or micro-CT. And whether it's in the cortical bone or in this case, I'm looking in the epiphyseal end of the femur of the long bone here. So you can kind of see this classic shape of the trabecular bone in this model. And what I want you to take away is that if you just look at the males between 3 and 5 months of age, you start to see significant evidence of reduced bone quality by way of reduced trabecular thickness. You also see this in the females. And you also see reductions in bone mineral density as well, both in the cortical and in the trabecular bone, both in males and in females. There is some distinction to be made, particularly on the bone formation and development side between the two genders. But if you're interested in that, you're going to have to ask me afterwards or wait till the paper comes out, hopefully. But just as a way of confirmation, if you look at kind of standard mechanical competency metrics like stiffness and max force and ultimate tensile stress in a 4-point bending assay, the sickle mouse manifests sickle bone disease, right? And so here's where I want to start to make a transition, okay? So we're not the first to speculate that S1P plays a role in bone homeostasis. We are one of the first to start looking at its potential role as a kind of a mediator of sickle bone disease. But we're trying to put 2 and 2 together here, and that if S1P is dysregulated and S1P plays a role in immune homeostasis, then S1P may be a molecular basis of how sickle bone disease comes about. Now, one of the things that we wanted to try to tease out is whether or not the manifestation of altered bone microarchitecture is a result of changes on the anabolic side, where bone gets formed by these osteoblastic cells, or it's on the catabolic side, where bone essentially gets broken down for the purposes of subsequent remodeling by these osteoclastic cells. Both can respond to S1P, both can secrete S1P for that matter. But what this is indicating is that there's this belief that S1P derived from osteoclastic cells can increase the proliferation and bone forming potential of these bone cells. So much so that if you knock out the cathepsin K protease, which again is the protease that really the osteoclasts are using to break down bone, and you screw the osteoclasts up, as a consequence, one of the things they do is they begin to upregulate S1P, and you can see in that osteoclast-specific cathepsin K knockout, you can actually see bone volume go up and not down, okay? So anyway, that was published. It's not our work. But what we did, we haven't done the osteoclast-specific knockout yet, but what we did was we did what's called a mouse marrow chimera experiment, where we took bone marrow, either from a normal AA donor or an SS donor, and we lethally irradiated a mouse, and in this case, we lethally irradiated either a C57 black cyst wild type or a cathepsin K knockout mouse, right? So in this mouse, this is a global knockout of cathepsin K, so all of its stromal cells, these they don't have cathepsin K, right? So essentially, by transplanting in SS marrow, we're giving these mice sickle cell disease, right? Because the sickle cell hematopoietic cells is essentially replacing the host's hematopoietic immune cells and immune system. And when you do this, essentially what you see is that you can give a C57 wild type mouse some of the hallmark features of sickle bone disease, and that the volume of their bone goes down. But if you do this in a cathepsin K knockout animal that presumably, and we had reason to believe that the changes in S1P concentration and other sphingolipids did ultimately change cathepsin activity in these sickle mice, right? But that change in cathepsin activity is not purely mediated in the cells that come from the bone marrow, like the osteoclast. The stromal compartment also is playing a role. And the reason we know this is that in the cathepsin K knockout, when we transplant the sickle mouse marrow, we still see indications of sickle bone disease in a way that is not recoverable, presumably, by knocking out some of that osteoclastic, or at least that in this cathepsin K proteolytic activity, right? Point being, we began to believe that, okay, well, maybe S1P is screwing things up in terms of messing up bone homeostasis on the anabolic side as well. And this is where, I think it relates to some of our work in stem cells. So here's just some histological indication that confirms that, when you look at just the pattern of how bone is forming in these animals, you can see that the transition between kind of the chondrocytic precursor stage and the kind of fully differentiated osteoblastic stage is delayed, and that, makes some sense. But here's a takeaway. What we did was we isolated mesenchymal stem cells, you might call them, or mesenchymal stromal cells from sickle animals. And we took them back to our mass spec profiling protocol and discovered that if you look at the plasma membrane contents of MSCs that are essentially isolated from sickle cell mice, some of the sphingolipids in those cells are permanently and very significantly altered in the disease state in a way that is preserved in culture. And we do think that this is partly a contributing factor in sickle bone disease because, sphingosine is the precursor to S1P as an example. And so if you essentially increase the pool of sphingosine in an MSC by 100-fold, it's going to have some consequences on S1P signaling and subsequently the trafficking and differentiation of bone cells. But that's kind of, some of the studies that are ongoing in our laboratory. But what about this idea? What about this idea that the plasma membrane lipids and their concentration profile may be an indication of the fitness of the MSC to do its role in maintaining and mediating bone formation? That really intrigued us. And it got us thinking about a potential role of profiling for membrane lipid composition, particularly in the sphingolipid family, for the purposes of cell manufacturing. So stay with me for a second. So I'm still going to talk about lipids, but now I'm going to transition to think about cell manufacturing for a minute. So mesenchymal stem cells or mesenchymal stromal cells are envisioned as therapy for a wide range of applications because they have pleiotropic pro-healing effects in their ability to modulate inflammation, their ability to modulate neurogenesis, the way in which the cell death mechanisms are regulated in these cells can also play a significant role in kind of modulating immune response. They can secrete trophic factors and so on. They could potentially be used as inducers of angiogenesis, but the potency with which a MSC donor lot is really mediating any of these pro-healing effects is variable. And and that's fairly well known. And so what we need is a way of predicting and preserving the high potency state of MSCs in the hopes of arriving at FDA-approved mesenchymal stem cell-based therapies, right? So essentially, that is kind of the motivation behind this emergent cell manufacturing emphasis that's happening at Georgia Tech, Emory, and various other institutions and countries for that matter. And now even as clinical trial results continue to kind of come out, we're beginning to see that some of these anticipatable problems with donor-to-donor variability and tissue sourcing and appropriate handling conditions, which kind of brings me back to some of the work that I used to do in rotating bioreactors, as Gordana mentioned. All these things become, important questions in our ability to determine, what makes a good MSC, and can we come up with quality attributes where a manufacturer of a mesenchymal stem cell therapy can actually measure something to predict and monitor and adjust the potency of their therapeutic. And we're particularly interested in the immune modulatory capacity of mesenchymal stem cells. And so that's what I'm going to kind of focus on here. Now, my colleague Chris Roy, who runs the Center for Cell Manufacturing at Georgia Tech, is an NSF-sponsored ERC. Carolyn Yago, who also runs our Marcus Center for Cell Characterization and Manufacturing. I was a bit player on a paper that was published reasonably recently in bioRxiv, where we did some single-cell RNA-seq of mesenchymal stromal cells from multiple donors. And this is a complex figure to kind of digest. There's several of them in the paper. It was a really interesting paper. I would encourage you to look at it if you're interested in this topic. But I just want you to take away mainly here that the single-cell RNA-seq profiles of cells that we used, where in particular we compared bone marrow cells to umbilical cord tissue-derived MSCs, so two kind of accepted sources of MSCs, and we kind of isolated them from multiple donors. And we looked at some of the genes that begin to distinguish these two tissue sources. A lot of network, some of the networks that were most highly regulated that distinguished these two tissue sources ended up relating to lipid metabolism. I'll just show you one that may catch your eye. So sphingosine kinase 1 is one of the two isoforms of the enzyme that converts sphingosine to S1P, right? So right off the bat, in addition to several other things that you can kind of see on this, kind of fancy looking diagram, we had reason to believe that looking at sphingolipids might be a way for us to begin to identify either attributes that can help successfully identify the source of the cells, or perhaps distinguish different states of potency in the cells, right? And so protocols do exist. So this is just a paper I took from a relatively recent study in Nature Chemical Biology that looked at essentially, which is not necessarily important to appreciate what I'm about to tell you, but this particular paper was looking at the plasma membrane leaflet distribution of lipids, right? So, this is, a ceramide, PE, PI, PS, sphingomyelin is something, should be something familiar. This is also a sphingolipid. So ceramide and sphingomyelin are the two dominant classes of sphingolipids in the plasma membrane. The rest of them, I think, should be familiar to you in terms of lipid, families of in the plasma membrane. And here's the upshot. The upshot is that there's a lot of sphingomyelin in the outer leaflet of the plasma membrane. And the other thing to take away is that the mass spec protocols for generating the lipid profiles of different classes of lipids and all the different structural variations on these lipids are very robust. So meaning, it's a little hard to kind of absorb, but I think I mentioned to you that sphingomyelin is kind of dually acylated. There's that base sphingosine, and then there's a fatty acyl chain, and it can vary in length. So 42 is the sum of the carbons in the main long chain base and the fatty acyl chain. 2 is the number of double bonds that are in that fatty acyl chain. And this other 2 is the number of hydroxyls. But the point is, we need this complex of nomenclature in order to distinguish all the different structural variants within these different species of single myelin. So my point is that using mass spec, we can distinguish a really large range of individual molecules in the family of sphingolipids, all of which could theoretically become the basis of some type of a biomarker for the origin or state of potency in a mesenchymal stem cell. And those things exist. And other reasons to or other motivations for us to look at the family of sphingolipids as a way of finding quality control attributes for mesenchymal stem cell based therapies relate to the fact that, say, the ratio of sphingosine and ceramide, is a very strong predictor of different mechanisms of cell death, ranging from, apoptosis, to, other forms of programmed cell death as well, particularly autophagy. And obviously, I've talked about S1P. I talked about it in the context of sickle bone disease, but if you look at S1P literature, what you find most significantly represented in the literature is the role that S1P plays in the trafficking of T cells and other immune cells. In addition, we've done some work also identifying how S1P can play a role in determining the polarization states and macrophages and the selective recruitment of different populations of monocytes from the blood. And these structural proteins like the sphingomyelins and the ceramides, they play a very significant role in things like the biogenesis of extracellular vesicles. And many of us are interested in exosomes and their biology and their potential uses as therapeutics. And so point being is that we felt like we were barking up the right tree to look at membrane sphingolipids as a way of finding and looking for quality control attributes of MSCs. So what we did was, in partnership with with CMAT and the Marcus Center at Georgia Tech Emory, we were able to get multiple donor lines from multiple tissue sources, right? So we had essentially from adipose-derived sources and donors, from bone marrow-derived sources and donors, and umbilical cord tissue-derived sources and donors, mesenchymal stromal cells from these sources that were grown in culture, passaged to P1, and then profiled using the workflows that we had developed for red blood cells previously, and motivated by a desire to do some work that was relevant to sickle cell disease in West Africa. We were able to leverage that in a way that helped us, find or at least, look for ways to understand, immune modulatory potency in MSCs by measuring these different classes of lipids. And again, I'm not going to ask you to memorize all these names, but these are other sphingolipids that we had learned how to measure. And a couple of takeaways that I'll, somewhat zoom through is this idea that, take a look at, cord tissue, adipose, and bone marrow derived MSCs. And there's a lot to look at on this slide, but if you just look at the, kind of the blue pool here of ceramides, you can see that there are reasonably robust, changes in the relative distribution of these structural sphingolipids based on tissue source. That's one of the takeaways, right? Now, in the dotted lines, you're actually looking at the profile of some of those long chain base lipids. And that's the family of lipids where you tend to find those bioactive lipids where you have kind of a receptor ligand regulation that's actually happening in different types of cells. And there again, you can see patterns that are reflective, broadly speaking, of tissue source. And so the complex sphingolipids, in addition to the long chain based sphingolipids from MSCs, can, we believe, accurately reflect and maybe predict the origin source, which gave us some confidence. And, to the extent that you believe in the idea that the cells from a donor maintain, some, gender relevant identity, right? Do the cells have, some type of, biological sex that's retained in the stromal cells from the bone marrow? Well, we actually had some male donors and female donors, and you profile the sphingolipid, kind of concentrations. And in this case, I would say don't look at the ceramide pool, but if you look at the sphingomyelin in this dark, navy, you can see significant changes. Basically, all I'm saying is that, our data thus far suggests to us that there are some differences in the plasma membrane, and particularly at the outer leaflet, because remember, most of the sphingolipids are in the outer leaflet that are directly exposed to kind of our culture conditions. There are some sex-specific differences that we've been able to detect so far that may be relevant for us to kind of continue to study. Now, back to immune modulatory potency, and then I'll kind of make another brief transition and before I quit for today. But, from our industrial partner, Rooster Bio, we were able to obtain a collection of about 10 bone marrow derived mesenchymal stem cell lines that they have developed from different donors with different levels of immune modulatory potency. And that immune modulatory potency was determined by their performance in a number of in vitro assays that are kind of outlined here from IDO, TIMP1 and 2, HGF, IL-8, VEGF. We're going to pay particular attention to this IDO assay. So this is indoleamine 2,3-deoxygenase. It's an intracellular enzyme responsible for the conversion of tryptophan to kynurenine. And you can essentially, because there's so much evidence supporting the idea that mesenchymal stem cells are actually using this particular enzyme's activity as a way of regulating and promoting the kind of the resolution of inflammation, you can use it as an in vitro assay to essentially stratify different donors by immune modulatory potency based on their relative IDO activity level. And so that's what we did. So we had 10 donors. And so we separated donors by IDO, whether those donors are above the mean IDO value or below the mean IDO value. If they're above the mean IDO value, they're high IDO, low, obviously. And we generated our mass spec protocols. And so, suffice it to say that if you generate a mass spec based profile of membrane sphingolipids, and you get a number of different species that are contributing to the variance between high and low IDO groups. And I should say that assay that I showed you is done in the presence of interferon gamma. These cells are not stimulated with interferon gamma. So in this case, without having to stimulate with any type of pro-inflammatory cytokine, the underlying sphingolipid profile between cells that are high IDO and presumably high immune modulatory potency and low IDO and presumably low immune modulatory potency is there's a significant amount of variance here between the high and low IDO profiles. And here's some of the actual molecules, and I can go over the nomenclature at the end if you want to know what some of these are, but suffice to say that there are some differences. Once you stimulate with interferon gamma now, where where these changes in IDO activity actually manifest themselves after stimulation with these cytokines, you still see the a variance between the high and low IDO profiles, and some of the same metabolites that were present in the unstimulated case are also contributing to the variance between a presumed high and low potency state of MSCs after stimulation with pro-inflammatory cytokines, which was important for us. And you can kind of see this here where, if I look at in this least squares discriminant analysis, all the unstimulated and the stimulated profiles together, and kind of reduce the dimensionality so I can project it here, is essentially the low IDO groups are more like each other than either of the unstimulated or interferon stimulated high IDO states, right? And so while high IDO stimulated and unstimulated may differ, both differ from the lower potency states, which gave us confidence that some of the metabolites that we're measuring in these profiles could, kind of form a basis for us to be able to predict the state of potency without having to generate the entire profile. And here are, this C16 ceramide is where there's 16 carbons in the acyl chain. 24,1 ceramide means there's 24 carbons in the acyl chain and 1 double bond. So these are particular metabolites where you can kind of see some of the changes between, states of high and low potency. And maybe I'll end on this note. There are changes, you can do correlation-based analysis of these profiles in order to infer what specific metabolic changes might be occurring here. And suffice to say that if you look at the correspondence between sphingomyelin and ceramide, that's where you begin to see that the high potency states are different. We can use tracer studies by giving a fluorescently labeled sphingomyelin molecule, and essentially those cells take it up, convert it to something else, like a ceramide species that is also fluorescently labeled. And that's kind of like an activity assay, so to speak. And, interestingly enough, if you use this as a surrogate model for sphingomyelinase activity using the tracer study, you actually see that the sphingomyelinase reaction, among all the different reactions that are happening in the kind of network of sphingolipids, is significantly increased in the high potency state. And lastly, second to lastly, you can actually augment the sphingomyelinase activity by taking a bacterial form of the enzyme and just stimulating the cells in culture. And the good news there is that both for high potency states and low potency states, the stimulation with bacterial sphingomyelinase tends to increase the immune modulatory potency of the cells. And I said second to lastly because I have to show you this or else I'll be really sad. Several slides that I'm not going to show you essentially are saying that exosome production is higher in sickle cell disease. Yes, I went back to sickle cell disease and then tried to take you back to cell manufacturing. That's because sphingomyelinase plays a role in the biogenesis of exosomes. And you can take mesenchymal stem cells and, I have a color wheel here. Oh, sorry. I don't know, my computer didn't like that slide. But the point is that you can, maybe I'll just end it here. Stimulation with bacterial sphingomyelinase in culture significantly increases the production of exosomes from mesenchymal stem cells. And we kind of predicted that on the basis of insights that we gained through the study of sickle cell disease. And maybe just one last little piece here that we're really excited about. If this, one of the things we were interested in is when you put MSCs in a three-dimensional hydrogel, in this case, PEG-4 maleimide is a particular formulation of a PEG-based gel that Andres Garcia here at Georgia Tech has really popularized. One of the things that we're really excited about is when you engineer sphingomyelinase activity into a three-dimensional context using this PEG-4 maleimide system, you can basically double or triple the number of exosomes that are produced by mesenchymal stem cells. And if you correlate the production of some of these immune modulatory exosomes with assays like IDO that predict the in vivo immune modulatory potency of these cells, you see essentially an associated increase in the immune modulatory potency of the cell, which may mean that some of our engineered biomaterials may be mediating effects in modulating immunity in different contexts of injury by the degree to which some of the, a cellular payload is able to kind of generate and deliver extracellular vesicles. Okay, so I'm gonna kind of skip a lot and maybe cut to the chase, where for some reason my thank you slide got deleted. My donors or my my funding source is gonna be really unhappy about that. I don't know what happened. Anyway, so I'll have to end it there. I just want to acknowledge some collaborations with the Roy Lab and the Marcus Center at Georgia Tech, in addition to Andres Garcia that I think I mentioned at the outset. And so I'll stop there and take any questions that you guys may have. So thank you. Rodana, I can't hear you if you're, I can.
Yeah, okay, thank you. A wonderful talk, a lot of exciting science. So let's try to answer a few questions, and then you kindly offer to answer some of them offline later. So before we do that, just to remind you that we will have 3 more tissue talks before taking a little break during the summer, and then picking up again in early September. So let's go back to Ed and questions. Joseph Freeman is pointing at the 60-minute story about the gene therapy to cure sickle cell disease. And he was wondering if you can comment on the effectiveness and cost of gene therapy of sickle cell disease.
I can't really comment on the cost. I think that the last time I looked, I have a student who went to the dark side and went to go work at McKinsey and became a consultant, but now he's working in a kind of a venture area, bio venture area in San Francisco, and he's been monitoring some of the up and coming, new therapeutics in the sickle cell treatment space. And I can say, efficacy wise, there are any number of actual curative therapies based partially in gene therapy that are really, really exciting. I have no idea about the costs. And I actually didn't see that particular 60-minute story, but I just,
cost is high.
Yeah. But I think one of the things that I just want to kind of plant a flag in, though, because I actually encounter this a lot in, kind of on study sections in particular that are, supporting work in this area. Yes, the advent of curative therapies based in cell and gene therapy is on the horizon, but we still very much need therapeutics to address kind of adult manifestations of disease. And that's partly the motivation in the strategies that we've taken.
Okay. A question from Kent Leach, a wonderful talk. Could you comment on the potential role of bisphosphonates that are, as we know, clinically used to slow down bone resorption in some of the models that you described today?
So that's a great question. And one of the things I didn't show is that we didn't use bisphosphonates, but we did use a cathepsin K inhibitor molecule, meaning like we tried to block some of the osteoclastic activity in the sickle mice to see if we could ameliorate some of the deterioration in bone quality. And one of the things we showed is that if you just try to block the osteoclastic activity exclusively, you can't really effectively, you can't recover the bone loss. Mm-hmm. Right? So you need, and I think that's partly because the dysregulation that's happening in sickle cell disease in particular is happening both on the catabolic bone breakdown and anabolic side. And the exciting thing to think about coming up with therapies that are treating sickle cell disease based on changes in lipid metabolism is that we believe that those changes are happening upstream of both the dysregulated bone catabolism and the dysregulation in kind of the catabolic and kind of anabolic coupling that happens in the bone themselves. And so we think it might be a much more effective therapeutic. And we do have some data that I didn't show that if you take a molecule that actually blocks sphingomyelinase, which is one of the reactions we identified as being kind of upstream of a lot of the inflammatory changes that are happening in sickle cell, you do get a significant recovery in bone volume. So we're hoping to continue that work.
Okay. So the one question maybe to just close by answering this question, Joanna Krzyspiak, she is asking if the composition of the lipid membrane of MSCs correlates to some extent to how successful the transplant treatment is because of the impact of lipids on exosome, on extracellular vesicles quality.
Right, so that is where I was going. We don't have direct data in support of that idea, but we do think it's true in that, number one, most of the sphingolipids that are present in the plasma membrane of the cell are on the outer leaflet and play a role in both the biophysical properties of the membrane and in some of the immune interactions that those cells have. Number two, we have been able to show that if you look at things like cell death mechanisms and apoptotic assays, and if you look at assays where you're actually measuring the production and secretion of exosomes, that there is a correlation between the level of underlying sphingomyelinase activity and ceramide generation and the number of exosomal particles that are generated. And so I believe that it's reasonable to presume that if I start out in a state of potency with a high distribution of ceramides, reflecting a higher kind of activity level of things like sphingomyelinase, that those cells will be more prone to some type of inflammation-induced cell death mechanism, and those cells will much more robustly secrete extracellular vesicles when they are placed in a stressed injury environment or some diseased tissue. Those are things that we believe. We have some small evidences in support of that idea, but I think much more needs to be understood.
It may be there is one more question that's a sort of remotely relevant. It's a really good question from Hannah Childs. Is there a relationship between the level of adipogenesis in the marrow space and S1P on the equilibrium, on the osteoblast behavior in the bone, intrabrachial bone?
So this may be a roundabout way to answer. I think the answer is yes, but so I'm ashamed to say, I told you I was experimenting on you guys to a certain extent, but there was a third part of my talk that I didn't even get to, that we are looking not at bone, but we're looking at muscle. And one of the kind of hallmark pathological features in something like volumetric, essentially a muscle defect that doesn't heal, is that you start to get fatty infiltration after the injury, and fibrosis and scarring that ultimately prevents a return to function in the muscle. And what we've done is that we've essentially reproduced a subcritical defect in the muscle and a critical defect in the muscle. And then we did lipid profiling, and particularly we looked for things like S1P, and we tried to see whether you can see correlations between things like fatty streaks and fatty infiltration and presumably some level of adipogenesis, and the concentrations of some of the pro-inflammatory lipids that we are measuring at the threshold between a subcritical defect and a critical size defect. So all of that is to say, the work that I didn't get to show you is that we believe that if you compare a critical and a subcritical defect, you do see significant differences in the concentrations of some of these pro-inflammatory sphingolipids. And at this point, I can't prove a cause, but those pro-inflammatory lipids do correlate with some of the pathological features like adipogenesis that are occurring in the muscle. And so whether it, and we feel like that essential story and molecular mechanism would play itself out in bone and other types of tissues just as, kind of easily as it would in muscle.
Thank you so much. You ended up with a lot of unanswered questions, but we will send you the information, and hopefully you can respond to those too. Thank you again for being with us today. It was really wonderful, and thanks everyone for attending Tissue Talks, and see you next Wednesday.
Thank you.
Bye.